welcome everybody to our last Sci Cafe of the season, I guess we're calling it, before we end the academic year. And if you have any ideas for Sci Cafe topics or anything you really want to know starting in the fall, please do let us know. Today's topic is on food security. Before we get to the panelists, I just want to remind you of the structure of how these Sci Cafes work. Each of the panelists is going to introduce themselves, introduce their area of research. I will ask a few questions as the moderator to kind of place them into a box, so to speak, to categorize their expertise so you know what they're studying. But the fun part of the Sci Cafe is always when you get a chance to ask questions. So that could be on Facebook Live, that could be here in person. We have this nice microphone that we can toss around. So please, as we go along, be thinking of your questions. And again, the idea here is to connect the research that happens at KAUST to the bigger picture, to how it impacts you, to how it impacts the community, to how it impacts the world. So please do be thinking about your questions. I have two items I need to take care of before we start. One is to chastise a few of the faculty who, instead of being here, may be working last minute on their CRG proposals. I just wanted to get that little jab in. And second, I wanted to thank the Plant Science Workshop, who I guess is here and sitting mostly in the front row, it looks like. These are 12 students from around the world who have been visiting KAUST for 2 weeks, right? And so they're getting a taste of plant science, and hopefully we'll have some questions to ask our panelists when it's time for the questions from the audience. So without any further ado, I want to let each of the panelists introduce themselves. Rod, if you'll start. And again, it's just, how did you come to this topic of food security? What kind of research do you do in that area?
Okay, thank you. Thank you, John. So my name is Rod Wing. I'm a new professor here at KAUST. I joined here February 1st, coming from the University of Arizona. I'm a genome biologist. I've been running what's called the Arizona Genomics Institute for the last 18 years, and we've had a big impact on plant genomics and food security. We were the first lab to sequence the rice genome back in 2005, and we've gone on since then to develop a number of biology projects revolved around world food security. One of the big things I'm very interested in is trying to understand natural variation in cultivated rice as well as its wild relatives. So I'll talk a little bit later about how we're going to make this pretty ugly looking plant look a little bit like this guy in a little bit.
Very cool, thank you. Ikram?
I'm Ikram, so I joined KAUST a year and a half ago. I come from the Netherlands, I call it the Greenland, and now I'm in the desert, and it's a beautiful place to study food security with a special focus on this beautiful plant, which is date plant, which is a very iconic plant of the desert, and my interest is mainly developmental biology, and I would like to implement how plants have acquired adaptive strategy to grow in desert conditions, using, as I said, the date palm as a model system.
Very cool. Simon?
All right, my name is Simon Kreitinger. I'm an assistant professor, and I basically joined one day before Ikram, 1.5 years ago. And my background is in molecular biology, molecular genetics. And I somehow got drawn into plant science and food security. And my research area is on understanding how cereal crops like this wheat plant here, for example, adapt to stress. And we are particularly looking at disease resistance, because diseases in agriculture, and particularly diseases in cereal production, are extremely important. And I think we're going to talk about this a bit later. So.
All right, so that was the quick, mostly scientific version. What I'd like to ask about from each of you is, I know when we had a brief conversation preparing for this, I was fascinated as a chemist that I don't think many people out there understand how agricultural research works. So it's one thing to say you study the genetics of rice, it's another to say you study the date palm or you study wheat, but maybe each of you can say a little bit about how research in agriculture, in food, even your research, starts to translate. What is the time scale? What does that look like? how does study on, what did you call them? You had a fancy name for the wheat. There's certain types of xerotropes, zero.
I don't remember, but I can tell you what we do.
Anyways, I want to know how the study of the technical stuff translates to eventually being in a field in Iowa or hopefully in Saudi Arabia. So, Rod, maybe you want to start?
Okay, that's a great question. So, earlier I talked about trying to understand natural variation. So, one of the main crops that I work on is rice, and I have a joint appointment at the International Rice Research Institute in the Philippines, and there they have 130,000 accessions of rice that they've gathered from around the world. And these 130,000 accessions are adapted to multiple environments to grow rice in Asia, in Africa, and in South America, and North America, so on and so forth. So we were involved in a project where we've sequenced 3,000 of these accessions, diverse accessions, and it turns out that you can divide those 3,000 accessions into 15 what we call subpopulations or races that are adapted to different regions of the world. So one thing that we're trying to do now is grow these 3,000 accessions in multiple environments around the world, phenotype them to see how they grow, which ones grow better in which climates, which ecosystems, and then capture that information and map that back to these 3,000 accessions so that we can accelerate the breeding process. Our goal is to really try to generate new varieties of rice that are higher yielding, more nutritious, but also ones that have less of an environmental footprint, ones that require less water, fertilizer, pesticides, can grow on marginal lands like we have here in Saudi, and as well as have reduced greenhouse gas emissions. Paddy rice production can account for to 25 to 30% of all methane emissions released into the atmosphere. So there is natural variation that exists in rice that allow us to reduce those methane emissions significantly. And we're trying to capture that and breed that into new varieties.
So what you're saying is over millions and millions of years, nature produced all of these different varieties. Some of them grow, as you said, in different climates. You're trying to use that toolbox and use more modern genetic tools to quickly figure out, can I cross this one with this one to get something that'll grow in the desert in Saudi Arabia? Can I cross this one and this one? That's kind of the general strategy.
Yes.
And how long, roughly, do you think it takes from understanding, or what's the trajectory of trying to take that information to then become a crop that can be done at a commercial scale?
Oh, that's a really good question. So we're going to have about 10 billion, 3 more billion people on the planet by 2050.
Yeah.
Okay, so this is a problem that we have to solve in our lifetime. So how long does it take to do this accelerated breeding process? With the cultivated species, somewhere around maybe 5 years before we can get a nice cultivar out there that is what we would call green super rice.
Okay.
To take something like this is Rhiza corctata. This is a wild relative of rice that can grow in salt water. That's one reason why I came here to investigate this crop. It has a range from Myanmar to Pakistan, and one thing we're trying to do is we've sequenced its genome. We want to learn the molecular biology of salt tolerance by looking at the genome, and then we're taking 2 approaches. One is to cross corctata into cultivated rice to bring in those salt-loving genes and then essentially be able to grow rice in very, very brackish water. Or the alternative is to domesticate this guy, make it so it's the one that's producing rice. And we can do that with using gene editing methods, and also we could cross. You can cross both ways. You could cross the domesticated type into the wild type and the wild type into the domesticated. So that's going to take trajectory there. That's like probably, with gene editing, we might be able to get a prototype out within 5 years, but it's probably, it's a little bit longer, 10 to 15 years.
Got it. So just to make sure I put it into perspective, if you followed natural breeding, if you waited for each of your crosses, I mean, date palm's an extreme example, right? It's like 30 years, but it would take you a long time to wait to figure out where to go next. The
Yes.
gene-assisted approach, which is what you're talking about, allows you to look at the genetics and kind of accelerate the process.
Absolutely.
And the extreme version of that would be if you do genome editing, which brings a whole separate host of ethical issues, potentially.
Yes.
Okay, good. And how about you and Ikram in terms of, date palm takes a while to grow. So
Yes, date palm is the final goal, but what we are mainly focusing in model systems like Arabidopsis, which is a wheat plant,
Yes.
but also tomato, which is really always, it's a big target for diseases like botrytis, which is gray mold. So if you leave your tomato in the fridge, the next day you will find a lot of gray things, which is it costs 100 billions of economic loss.
Yes.
And so what we are trying to do is to design plants that grow better, but they are also more resistant.
Yeah.
And we do that by using different approaches, genome editing, molecular biology, cell biology, and we mainly focused on the root system, and I will show here. For example, if you think about the root system, which is this picture here, like a digital root, and so this was done by one of my students, and the root system, you can see them as a sensor. So they are an electrical system which is used to very interconnected with one another, and it's the organ which acquire nutrient from soil. So if you don't have a good root system, the plant will not grow properly. And what we are mainly focusing in understanding how to have better roots not only in models but also in crops. Using also the date palm to understand its adaptation to desert conditions, because what we found, for example, that the date palm roots, they don't grow like this in a mainly vertical manner, but they make roots that are horizontal. So if you go out now after the Sci Cafe and then you dig up a little bit on the date palm, you will find roots going up, and that's a very fascinating way of adaptation to desert conditions, because rainfalls are really scarce. So the plant is ready to assimilate as much water as possible to survive the desert condition. And this we wouldn't have been able to know it if we wouldn't have been looking carefully at the root system.
So what you're saying is the kind of imaging and more fundamental research.
Yes.
You're trying to understand even the biochemistry of how do these plants evolve to tolerate harsh environments.
Yeah, an example, for example, if you find a gene which is responsible of making the roots better or grow faster, so by looking, for example, at cell division and cell growth. Yeah. So if you can take this gene and put it in a plant and make it grow much faster or much longer, then you will have, the root will colonize a much larger surface area. So that means it will have a lot of nutrient, more water capacity. So you can think about the root as a hydraulic system, where you can have all your nutrients and then facilitate the growth in a much better way.
Yeah, so it's not necessarily something you're going to translate immediately, but in combination with your colleagues.
It's the beginning.
It's the beginning of understanding how we grow in these environments.
Indeed, yeah. Well, for example, when the DNA was discovered, so who would have thought that we would get that far? Yeah. So with all the discovery that has these consequences.
Very good. Yeah. Okay, and Simon, your focus again is on I liked your Vanna White impression there. That was solid. Your focus is on wheat rust in particular. Yes. So yeah. Why is that such an important thing to focus on, and how does that help translate to growing crops?
Yeah, so just to give you a little bit of context, so 50% of the calories that we consume are coming from cereals, like rice and wheat. Yep. So when we talk about food security, we have to talk about cereals. In that sense, that you can say it that way. Global food security to a certain extent depends on our success to grow grasses, yes. Right? And diseases destroy about 15% of the global cereal crop harvest every year. Now, 15% might not sound so impressive, but that would be enough food to feed an additional 1.5 billion people. So just imagine we could get rid of all the diseases that affect wheat or rice or corn. That would already make a big contribution to what Rock calls the 10 billion people challenge. So what we try to understand is really how disease resistance is controlled molecularly and genetically in wheat, for example. So for example, this wheat cultivar that I brought here is coming from South Africa, and it's a very special one because it has shown an excellent resistance to these fungal rust diseases, which are very devastating for the past 30 years. So we got this variety from the local breeders in South Africa, and sort of they asked us to study this resistance, which we are doing now. The problem here really is that wheat has a massive genome. The genome is 5 times larger than the human genome. So if you would print the genome of this beautiful plant in a book, the book would be 55 meters thick. Wow. And basically, we are looking for the right page in that book, right? So which gene is controlling this resistance? So it's an enormous challenge, but we're using genetics and molecular biology to do this. And then the question, how do we translate this into breeding? So we are currently seeing a shift from what we call, just trait-based or phenotype-based breeding to molecular breeding. So what we do is that once we know which gene, for example, controls the disease resistance, or it could be any trait, the drought tolerance or salt tolerance, you can design a molecular probe or tag on that gene, and that can be used in breeding programs with molecular assays. So instead of looking at the plants and judging them based on how they look like, the breeders can then specifically follow those traits and genes in their breeding programs using these molecular tags. So it's kind of we're in a constant exchange with public and private breeding institutes to basically give back that information so that they can integrate this into their breeding programs.
Yeah. Very cool. Thank you. I'm shocked to hear that it's 5 times the human genome.
Yeah, no, it's big. It doesn't look like this, no?
It doesn't look that complex, but... all right, so I want to open it up to questions, but I want to just make sure to package what you heard. I like how Rod phrased it. There's a 10 billion people problem in 2050?
2050,
2050. So essentially what these 3 faculty and others at KAUST and around the world are doing is trying to find any improvements or any efficiencies in agriculture to help us achieve the additional food we need and potentially address the distribution issue of getting it to those 10 billion people. So that's kind of a twofold problem. It's reducing disease, it's improving the roots, it's improving the genetics of the rice, but it's also being able to grow in areas where you can't currently grow. So I think when we talked earlier, it's this very interesting challenge when it comes to food security. If I can grow all over the world, that really helps to solve the problem. If I have large regions that are arid, then I have a distribution problem and I have a few food security problems. So I think everything that they're doing aims towards those goals. So what questions do you have potentially about cereals, which are not going away anytime soon, about agriculture? Ask them about the Atkins diet or the paleo diet or whatever the fad is, and they'll give you the scientific explanation. What do you want to know? I have a nice, yes, right there in the back.
One of the biggest challenges that we have as scientists or academics is that we need to take our discoveries into society. I will give you the example of the golden rice that was described in 2000, and I think it was just approved last year, after 18 years, to be commercialized. You can find the best genome. What are the challenges after that to actually, maybe it will be more than to 2050. What's the challenge that you have after getting the right genome, the right gene, and the right phenotype?
Got it. Who wants to tackle it, Rod?
Yeah, I can. I mean, as I explained earlier, I mean, the different tools how you can translate it, and I think you were particularly mentioning, genetic modification, of course, and there we face issues with acceptance. But really, I mean, in our case, the biggest part of what we do in terms of translation is what we call molecular breeding, where we basically assist breeders with these molecular tags to be quicker, faster, and more precise in their breeding programs. So this is a GM-free approach, right? And this is what basically all breeding programs use around the world. So they shift from, as I just said, really looking at plants in phenotype-based breeding, which is very difficult to do sometimes for certain traits, to molecular breeding, where they use a lot of molecular information to they make crosses, and within the crosses basically find the best combination of genes and the best plant they want to keep. Just to give you one more number, for example, to breed one new wheat cultivar, you start with 1 million plants. And that's a medium-sized breeding program. So from 1 million plants, at the end, you are left with one new cultivar, and you need to find the right combination of genes that gives you sort of the best output in the region you're working in. So, we could talk about GM, and there are issues with acceptance in certain regions, and but really it's important to understand that a big part of breeding is not about GM, it's not about genome editing, it's really also about molecular breeding, where we just use molecular technologies to assist regular breeding, basically.
Okay, without getting totally uncomfortable by talking about gene editing, maybe, because I think part of her question was about the policy side.
Yeah.
So irrespective of gene editing, I mean, there is then some sort of process for people to adopt this, for people to use the cultivars, for this to get implemented more broadly than just the science suggests, right? I mean, is there anything we can do to help tackle some of that?
Oh, definitely, yes. I mean, inform the public.
Yeah.
Go out to policymakers, but definitely go out to the public and inform them what genome editing is, and that it's not that different from creating natural variation or looking at natural variation, right? I mean, it's just, it's not much different from what we do in regular breeding, but I think it's really, we have to go out and talk, and that's what we do as well. We go out and talk to the public.
Yeah, we had this conversation when we were briefing, but whenever I give a talk, I don't say a single word about GM, and then the first question is a GM question. So you're the perfect audience. So it costs so much money to introduce one event. It's maybe $1 billion in the end to have one event released in commercial application. So one gene at a time is not really going to do it. So I mean, really, what we're focusing on is this natural variation. I mean, rice, the cereals have been around a long time. The Ariza, Poaceae, all the cereals, they share a common ancestor 50 million years ago. So these crops have been around. They've been tested. Our goal is to find combinations that will thrive anywhere and in a sustainable fashion. So that's what we're really working on. But so like for gene editing, that's another story. That's when you can actually go in there and modify like a single base, a single gene, and this is it depends on where you're from. In the States, it's not too big of an issue right now. But really, I think in Europe, it's still gene editing is just as bad as GMOs, right? So and I don't know the policy here in Saudi, but we're basically exploring this molecular toolkit, identifying these genes and getting them into the hands of breeders that can put together something as quickly as possible. Because it's really a crisis that we have. In 3 more billion people on the planet by 2050. That's a huge problem. A majority of that is going to be happening in Africa, Southeast Asia, and it's something that we have to contribute to and for kind of world stability and food security.
I mean, can I just add on this? I think, as Rob mentions, policies are unclear, but policies can also change pretty rapidly, right? And developing these tools takes time, and I think we have to have these tools ready, and then we're going to see which tools we can use and which ones we can't use, but we have to have them ready, right? I think that's the most important thing, because we don't know how the policies will be in 10 years from now, right? I think.
But so far at KAUST, we are using gene editing as a research tool.
Yes.
To complement the larger, more natural breeding programs or genome-assisted breeding programs, right? So I think I like your point, Simon. It's an evolving policy landscape, and we kind of need to be ready. What other questions are out there? Here, you can use the fancy, are you ready? Yeah.
Good shot.
Wow.
Oh, good. So tying into this whole policy conversation. So in the United States, Row 7 Seeds they're crossbreeding for flavor, and they're open sourcing their seeds so that once they come to the one that they like the best, they offer it to the public, to researchers, to everybody. So how could that work, or what's the difference from coming from where you are to that kind of an approach to get it out there to people to grow?
So basically, you're asking them to assess the Monsanto model versus the open source model, essentially. Yeah? Without throwing Monsanto under the bus.
I mean, I guess it's, there's the research that's,
Yeah.
the research that's coming, I guess that's being financed, I guess, is that where you're saying the financed version versus people who are, that's the discussion I'd like to have here.
Yeah. So I guess there's different, there's some
Why
that's
can't there be more open sourcing?
understood. So
Yeah, so I mean,
Thoughts
in rice, it's definitely an open source field, because I mean, we have these gene banks around the world, and you can write for seed and you can use it as much as you want. I mean, you have to sign these material transfer agreements, things like that, but it's definitely open source. And the thing is at least, so I come from a school through being trained by the National Science Foundation that, essentially all of our data has to be immediately released to the public. So we release all of our data immediately. Companies can use it, private, public institutions can use our data to make improvements in crops. So that is, our work is completely open source. And that's same for the wheat genomes, and pretty much all of the plant genome projects, essentially all the plant genome projects for crops that are very important are in the public databases. And it's really where people start making money on this type of stuff is that they'll make a discovery, and they'll say, okay, we've discovered something that's better than everything else, and then that starts these commercial biotech companies, so on and so forth, and then they can produce seed that outperforms everything else that's out there. So I don't know. I think there's room for both commercial and public enterprises to work together. But a company owning a critical food supply, I mean, that's not right. I mean, the rice feeds the poorest of the poor, and you need to be able to grow enough rice to feed the poorest of the poor, and so it's pretty hard to make money in rice. I think you can make money in wheat, definitely in corn, and most of that is a kind of more hybrid rice production that they do. But your flavor idea or your flavor concept is very interesting, to be able to, somebody grows this, discovers a great flavor and then releases it. So that's fantastic, and I will definitely encourage that.
very good.
Yeah.
Yeah. You want to toss or you got it?
Thank you. Yes, I have a question. You mentioned that the rice production produces a lot of methane.
Yes.
I believe it's from decomposition of the peat in the swamp. So it's a problem of oxygenation of the sediment, basically. So how is it possible to modify the plant to avoid methanogenesis?
So the methane is actually produced by the microbiome growing in the paddies, but then the methane is actually released through the rice plant. Okay, so there actually have been studies at UC Davis where they've looked at very natural variation of methane emissions through different rice plants, and they've actually seen a significant difference between one cultivar versus another. So the idea is to try to identify those genes and use that to accelerate the process so that we can get that into mega varieties that are grown around the world.
So essentially, some of the varieties start to make use of the methane before it leaves the rice plant. Is that
Well,
what
they're
you're
not
saying?
really using it, but they're passing it through their system, essentially. So, but there's other ways to control methane emissions. There's so much just agronomic practices can change things. I mean, a lot of the genetic engineering we do is for future crops and try to be more sustainable, but just like laser leveling a field can have a huge increase in rice production. And what they do in rice for methane emissions is it's called alternate wet dry AWD, where you essentially wet the field and drain it. And when you drain it, so you don't keep it flooded. and that also has a big effect on methane emissions. But the problem is, I mean, that will work on the industrial scale. But these small farms that are growing this stuff, they can't do. They just don't have the capabilities to do that. And most rice farming is on small farms.
Yes, Donald.
So you've talked about using the plant product, so the seeds of the plant for food. I guess one of the other problems that arises is what to do with the rest of the plant. And so, for instance, burning rice straw in China is a big problem for environmental conditions and so on. Is there much work being done on looking at how to use that as a material that can then be a source, for instance, of green chemicals or some other products, rather than simply burning it?
Yeah, I mean, I would definitely say yes. I mean, it's kind of changing its cell wall structure so it's easier to degrade. That's being done, a lot of that work's being funded by the Department of Energy in the United States. And so they have kind of bioenergy programs versus feedstock programs. And so, I mean, yeah, I mean, I just was recently in Colombia, South America, and I saw them burning the sugarcane fields. And it's just, it's crazy. I don't know how much field burning they do in Africa, but do you know? Or I know I got my PhD in Davis, and they grow rice around there, and they used to burn the rice paddies, every time they grow the rice. It was terrible. So, but there are definitely efforts to do that. We're not doing that here, but again, by looking at natural variation already, that we can extract that out of these cultivars.
What about for wheat and some of the other grains?
Well, I mean, what you try to do in wheat is to basically reduce, so get as much of the energy into the grain as possible. I mean, when you look at the traditional wheat cultivar, it's about double to 3 times the size of this one. But currently, I mean, the straw is not really used a lot for producing bioenergy because we use different plants like corn, for example, that are much more productive in terms of, look at these tillers here, they're pretty thin compared to corn. So no, there's actually not much you can do with this at the moment.
Got it.
Actually, I have to say that the winning one is still the date palm because you can use every bit of it for everything. So you can use the leaves to make purses, to make carpets. You can use the trunk to make your roof. Even the pit, you can grind it and have a date coffee with it. So every bit of it is really nicely and well used for commercial purposes.
Date coffee. We should
I
ask
have
Tim
it.
Hortons to
include
make
it.
some date coffee for us.
Yes.
Was that just a sponsor plug?
Oh,
Sci Cafe is
The
getting
cappuccino.
professional here. I mean, the new VP for research is here asking questions. I mean, this is a serious production. I like this. Any more out there? Yes.
Yes, hello. A big part of food security is not just giving enough calories to people to eat, but it's providing sort of nutritious food. So my question is, in terms of cereal research, are you doing things here at KAUST to improve nutrient contents of cereals, like with golden rice or biofortification of cereals?
Maybe give them, just because not all the audience knows the golden rice, so maybe update them on that.
Well, the golden rice was for to put in provitamin A into rice for children to eat, so they wouldn't. Vitamin A deficiency can cause blindness, and so the concept is the poorest of the poor, they need to eat something that they can get their vitamin A from rice. And but it's taking, like you said, 18 years for that to get actually publicly released. So that's biofortification. So my group specifically is not working on improving the nutrition, but by studying again this natural variation in collaboration with places like Erie, Africa rice, Simit in South America. Again, we're screening these thousands and thousands of accessions, and not only looking for growth in marginal soils, but also assaying the nutritional properties of all the rice that's there, and then capturing that, and then putting those into accelerated breeding programs as well. So that is definitely happening, but it takes a village. It takes a whole team to put this kind of things together. One scientist can't do everything, but we can. So for an example, one thing that I want to do at KAUST actually is I want to import these 3,000 accessions into KAUST and grow them here and phenotype them here in a completely different environment that other people are doing, and we could send those seeds for nutritional quality testing and many other things.
Well, I agree with your comment that it's not just about yield, it's also about quality. But as Rod said, there's 7 plant science faculty here, so we can't do everything. But quality, particularly in wheat breeding, is basically in the top 5 of the most important traits that breeding programs look at. So mineral content and vitamins. So it's done. So people look at quality.
Yeah, he's not here represented, but I believe Mark Tester is working on some work related to iron content, improving the iron content in wheat.
Iron content in wheat and rice.
Yeah. So I think there are some efforts here at KAUST, just not necessarily represented by this panel, but as Simon said, these are all things we're trying to tackle. What other questions are out there? Yes.
Just a very simple question. What are the main limitations, what are the main limiting factors for your research programs to be translated or to contribute to food security? Is it time? Is it funding? Is it the lack of a village? Or is it the lack of policies? And is it the same for all 3 of you?
I can start. For instance, in the case of date palm, it's just genetic limitation. The plants take 30 years to grow. If you want to do breeding, forget about it. We can start something, but then maybe the son of your PhD will follow if he continues on science. Then the genetic variation. So palms have male and female. So then you really have to select the plants that you want to cross to make sure that you will get the right fruits. So these are some of the limitations that mean that it's not impossible. So being in a place like here in KAUST with all the facilities that we have, so the idea that we are trying to do with all the plant faculties to design fast fit palms or plant in general, in which we can take advantages of the let's say the desert microbiomes, like to make plant grow better, take care of the desert to make them more tolerant to salinity and to drought, and then having hopefully one day we will have the desert become a paradise, a Greenland. But this will not be possible if we don't really put our hand together and have it as a consolidated effort. Of course, policies might be a problem when we start talking about GMO and all these kind of things, and that need mainly education. So we need to educate the public that GMO are not killing us. I get it's really amazing that you go to a supermarket and then you see labels, this apple has no DNA. As a biologist, that's really bad. So I think maybe already at the level of school, we have to educate our children that GMO are not going to kill you, for example, or introduce any diseases.
Well, I think, one other limitation that you often face is also convincing breeders that they should go back in what, for example, Rod described, looking at all old land races or even wild relatives and incorporate it into their breeding programs. Because often what happens is, breeders, they have worked with a certain set of material they know, a few, or, let's say a couple of dozen of different cultivars they cross, and that's what they know, that's the material they know and the material they like. So really convincing them to bring in something completely different and new can sometimes be challenging. But we also see that this is changing now. I think, breeders have realized that we really need all the different, all the variety, basically the natural variability that there is to make our crops better.
Yeah, I would say That's a good question. I think part of it is a little bit of a lack of funding, for sure, and so that's a funding pitch for here. So I'm involved with the plant science group here and some folks in the States. We're talking about trying to put together what's called a science and technology center in agro-ecosystem genomics, and the concept there is to not only look at the genetics of the crop plant, but also simultaneously look at the genetics of the microbiome, so that we can identify the combinations that are going to be more sustainable in the future. So that's a huge area, and we're looking at trying to work on rice, sorghum, maize, and wheat, and identify grand challenges related to those cropping systems, and then how they relate to yield, carbon cycling, greenhouse gases, and try to develop a model like an Earth system model that would include what we call genome association or genomic selection, and so that we can essentially predict what genotype, what land, what microbiome, what right combination that we can grow on 100,000 hectares to efficiently during that season in that ecosystem. So that kind of coordinated effort is something that we really need desperately.
I'm impressed with your restraint. He kind of put the softball up there and said, "What resources do you need?" So I'm going to follow up. Only Rod was the one who said we need more money. I mean, are there, maybe let me focus the question a little bit. In kingdom, because some of what we're trying to do is to have these things be translated in kingdom. So are there limitations in terms of the fields that are necessary to scale? Are there limitations in terms of partners? What sort of things, I mean, we have not a global audience, but a Sci Cafe audience here. What sort of things do we need to translate in kingdom in particular?
I mean, I can probably add on this because what I said, I mean, we are all doing basic science. Actually, I think none of us really does breeding, but we collaborate with, as I said, with public and private breeding institutes. And I think if you talk about the kingdom, what we need here really is breeding, I think, I mean, and one thing we try to do is bring in also breeding companies into the kingdom to visit the kingdom and see and probably establish collaboration. I mean, it's a huge market for them as well, right? I mean, 33 million people trying to push agriculture. So, I mean, I think it's really important that the basic science that we do here, and probably also the more applied science, can really go into a breeding program. That's the first step. We take our science, the discoveries, and that goes into a breeding program. And from there, it takes about 5 to 10 years until you get a final product, I think. And those 5 to 10 years at the moment, I think, the infrastructure for this is not existing. So, yes, of course, on our side, we need a field testing area on campus, but we also need an infrastructure to really get it bigger and translate it to, or give our discoveries to a breeding institution.
Got it.
And just another challenge that we're facing, for example, we don't have contact with the local farmer, for instance. So we don't know what problems they have. And then if we would have this contact, for instance, have access to farms for different crops, not only for wheat, for date palms, then at least we will get in touch what kind of problems the farmers are facing. How can we implement our basic research to help them to move further? And one example is the consortium that we have for to develop sensors to detect early infection by red palm weevil. So it's an initiative by KAUST. And one of the major challenges that we were facing sensors are being developed, but where to test them? So that was something that we are slowly now getting in touch with locals to be able to test all these beautiful devices that people from electrical engineering, from biology, and from many other disciplines within KAUST that we are trying to implement them.
Yeah. Just for those of you that don't know, we have a small collaboration with several entities, the Ministry of Environment, Water and Agriculture, and others, on red palm weevil, which is a huge pest for the date palms. And just to echo Ikram's comments. We I think, have some technology that could work. We're at the scale of we're now trying to scale it to Saudi Arabia. Do you have anything to add on that?
Well, I would just say, I mean, I haven't been here long enough to learn the lay of the land, but I do want to take a tour of Saudi, of the agricultural regions, and learn about what, I mean, we probably need a field tour, the whole team, to go out and figure out what's there, what we have to work with. And I think, like you said, breeding is critical, but I think I don't know if KAUST necessarily needs to have the breeding program here, but if there's a breeding institute, in the kingdom, would be fantastic. And we could translate our work to them in various field stations.
I don't think we need a breeding here at KAUST. I mean, I don't think we have the capacity.
Yeah.
And it's also, I don't think it's the mission of KAUST to do this. I mean, the mission is to get research and get it out and translate it. So I think we need something outside, that can make use of what we produce here.
Yeah.
Got time for one more question. Where are we go? Oh, there we go. Yes.
Okay, maybe just one comment and one question. So for Dr. Ikram, here in KAUST, we have more than 11,000 palm trees. So I can catch up with you afterwards, see if we can do something here. My question is, when we talk about food security, of course, rightfully so, the main focus is about feeding human being. My question is, and coupling that with the quality of the products, can we also focus on the biofuel and feeding livestock as a side result of those research?
So I have to give a shout out, because this is now the second Sci Cafe in a row. That's our director of campus services. Amar's asking the question. I'm just putting a challenge out there for more of the KAUST community to come and kind of participate in some of these things. So, I do encourage you to follow up, because he may be able to help you test some of these on the 11,000 palm trees that are here. But what do you have to say about the biofuels or saline agriculture or feed?
Well, I would say with respect to like forage crops, I mean, this is the same idea for being able to grow high-yielding forage crops that can be, grown in marginal environments, that can feed the animals, that for higher protein content. That's definitely part of this. This concept. Right now, we're focusing on cereals for human, but it applies directly to forage crops as well, for sure.
Yeah, no, I agree. I mean, of course, thinking about biofuel and growing crops for food. I mean, of course, there's a certain conflict there that is difficult to resolve. But I agree with Rod's comment. We have to go into, or what we really have to do is to grow or be able to unlock land that we can't currently use for agriculture, marginal land. And I think that's the key. Even if we are not as productive as we are on, better land, but if we can produce without having to put much irrigation into it or fertilizer into it, I think that's the key to do. And if there's one thing in this country, it's space, empty space. We have space for agriculture, we just have to unlock it.
Well, and that opens a can of worms that we don't have time to address now, which is tying back to some of our previous Sci Cafes on water usage, water security, because all of this starts to tie together, because if you don't do it efficiently, you can't grow on that land because you don't have the water to sustain it. So with that, I want to draw it to a close, but I want to highlight the in-kingdom piece that was brought up here. I do think that KAUST is working with NEOM. KAUST is working with the Ministry of Environment, Water, and Agriculture, and I think there is, in the last, I would say, year or so, there is a lot of increasing collaboration there to take some of the basic science that's happening, start to look at field trials, and start to look at scaling this technology. So hopefully, as we go through the next season of the Sci Cafe, we can start to indicate what sort of progress has been made there. So please, thank you for coming to all the events throughout this academic year. Please join us in September when we'll have the next Sci Cafe. And please thank our panelists for participating.
Thank you.